Several different types of COVID vaccines are available today. Of these, the long history of use of inactivated vaccines gives them advantages, such as a well-documented safety record, well-developed manufacturing processes, and the ability to present multiple viral proteins for immune recognition. Owing to a limited supply and the urgency to inoculate people against COVID-19, inactivated vaccines may be a relatively economical and effective method for producing safe vaccines. In this regard, scientists from China conducted Phase 1 and 2 clinical trials in 18 to 59-year-old adults for an inactivated vaccine called CONVAC in October 2020. 60 participants in the Phase 1 trial received a 5 or a 10 microgram per dose of either the CONVAC vaccine or a placebo on days 0 and 14. Subsequently, 500 Phase 2 trial participants received either a 5 or 10 microgram per dose of CONVAC or a placebo on days 0 and 14 or days 0 and 28. The primary endpoint in the Phase 1 trial was the proportion of participants experiencing adverse events within 28 days following the administration of each dose. In the Phase 2 trial, the primary endpoint was antibody response. The percentages of participants who experienced an adverse event or vaccination-related adverse event among those who received either a 5 or 10 microgram dose of CONVAC or a placebo were similar. No adverse event of grade 3 or higher was reported. CONVAC induced significant antibody response, with those on the day 0 to 28 regimen showing a higher immune response than those on the 0 to 14 regimen after receiving 2 doses. At 28 days after administration of the second dose on the day 0 to 28 regimen, 99.0% and 100.0% of participants who received either a 5 microgram or 10 microgram per dose of CONVAC respectively underwent seroconversion for neutralizing antibody to live virus, whereas only one placebo group participant underwent seroconversion. Geometric mean titers, or GMTs, of neutralizing antibody to live virus were 131.7 in those who received the 5 microgram dose, 110.7 in those who received the 10 microgram dose, and 2.0 in those who received the placebo. The GMT in participants who received the 5 micrograms per dose on the day 0 to 28 regimen was 2.65 times higher than that of the convalescent serum. The study shows that administering CONVAC at 5 micrograms per dose and 10 micrograms per dose induces robust immune responses in healthy adults. Based on these results, Phase 3 trials will begin shortly.